Thank you so much. Such a powerful panel. So many questions, so little time. But I think it was worth hearing all of those stories in full. And I think we heard many very concrete and direct implications from the examples from all of these panelists for clinical trial study design. We can take questions now, but I think I thank you for providing those. I hope everyone who is a clinical trialist has been taking good notes. I think there is a lot to be learned from this panel. So we'll go to questions.
Good afternoon. I'm Derek Robertson, Maryland Sickle Cell Disease Association, a member of the forum, a parent of 2 sons with sickle cell disease. First, I just want to thank Maria. You're an inspiration to every parent. Thank you. And Tesha, inspiration to me and to my sons. Thank you very much. So my question, Courtney, I think, or Dr. Fitzhugh, sorry, I think you mentioned that on the gene therapy trial you stopped hydroxyurea. I just wanted to know if that was, or Dr. Tisdale, if that's the case, why? And then I was just wondering, Dr. Tisdale, if NIH or any other agencies looking at educational materials for patients around the different types of techniques, because just like you, as a layperson, I hear about CRISPR, zinc finger, talons, prime, all of these different things. And how does that impact? What are the differences? And so not to necessarily answer that today, but if there's a plan for the future moving forward to get materials out so that we can just understand the process a little better. Thank you.
So Courtney punted to me the hydroxyurea question as well. So hydroxyurea is really the one drug we have for treating this disease and has a consequence of lowering the number of pain episodes per year quite substantially. But it also reduces the number of bone marrow stem cells that we can get from a patient. So in order to increase the likelihood that we reach the target and have a good dose, we have to stop hydroxyurea. And so what we do instead is to start a chronic transfusion regimen, which is equally effective for most of the endpoints that we measure in clinical trials with sickle cell disease, and can also serve to sort of settle down the bone marrow. So we think we get a double benefit from that. Stopping the drug allows us to get a higher dose, and doing exchange transfusions before the harvest potentially gives us a better product, that's less prone to the downstream complications and processing and all that. So, the second question was educational materials, right? So NHLBI is working.
I'll just add to that once, because I was thinking if you could say why you have chosen CRISPR-Cas9 as opposed to maybe zinc finger and some of the others as well.
Well, one of the reasons is access. So some of the technologies are protected by company interests that don't allow us to get as ready access. CRISPR is a little bit easier for us to access. They seem to be pretty equivalent in terms of efficiency. They all have their pluses and minuses, but we chose CRISPR because it just was the most accessible and worked really well right out the gates. In terms of education, NHLBI is working on educational material. Julie Cantor, also in the audience here, is also working on educational material to try and, make this digestible. I mean, some of this, is complicated. We'd ultimately like to make it simpler so it's more exportable, but for now we're stuck with what we have, and we have to do a better job of educating. So.
I have a question, if we have a couple of minutes, for any of the panelists. And so I think we've heard many specific suggestions, or at least implied suggestions, for things that should have been concluded in informed consent and weren't. But we also heard from Dr. Fitzhugh about the difficulties with really not just understanding informed consent, but really understanding informed consent. I think many patients who are participants really are experts in their own disease and understand the risks and become experts in the science as well. But I think what we know from from many empirical studies is that while people may understand that there are risks of death and risks of severe morbidity from participating in clinical trials, the issue is the extent to which you believe you're in that category of population that is going to be subject to those risks. And it's very difficult for people to sort of reconcile those two ideas. So given that, but also given the really great suggestions for informed consent, I just wonder how people, if people have suggestions for sort of overcoming this cognitive dissonance.
So one thing I could say is that a gene therapy consent form runs 22 pages and a tiny single space type. It's crazy to try to explain this in one sitting, even to a postgraduate, highly educated person. And so I think there need to be many conversations about participating in clinical trials and gene therapy in particular, and I think people have to know that they maintain control. They can't. We say you're a volunteer. You can stop being in this anytime up to the time the cells get infused. After they're infused, those corrected cells, you've got them. We want to follow you, and we just have to be partners. And I think the communication and trust and partnership doesn't develop over one consent form. It develops over days to weeks at least, and hopefully longer.
I completely understand, and it takes multiple times and usually weeks to really explain to them. I usually ask them to read the consent by themselves in their own time and write down any questions that they have. I ask them to go and talk to their own doctor, to talk to their family, to people at their church to pray about it, and also to talk to other patients. I'm glad that was brought up in the study. I always ask them to talk to patients who have gone through transplant, because they can really explain better to me what they're actually going to experience, not only where the transplant worked, but also when it hasn't. And that's been really helpful. And the patients are always happy to talk to the upcoming patients about the trials.
I just want to ask to Tisha, because you said that you were able to reach out to people that made themselves available, and then you were also able to talk to others who had direct experience. Would you have been, or did you, would you have been willing to, or did you actually sort of talk to people about sort of the real talk on informed consent?
Absolutely. So I actually brought the initial consent that I signed. So this was emailed to me literally a year before I decided to sign it, so that I could really pore over what's in the informed consent, come to my own conclusions about it. And then I had several conversations with the research nurses, and then I had to have a meeting with my family and friends. We actually sat down in a boardroom, so that, because when you have a support system, they need to know what to be aware of. So I believe that you have to not only just go through this, and you have to literally pore over it, because a lot of it isn't in layman's terms. So you have to make your own decision about it and read about it, but also just continue that conversation with the doctors, Dr. Tisdale, Dr. Shea, that never stopped once I became enrolled. Even after the busulfan, and I'm feeling all of the things that I'm feeling, I could still talk to them about what I was feeling.
Yes, I think it would be necessary to shift the notion from consent form as a point event to consent process. And from that point of view, in our experience, it would have been really necessary not only the suggestion of the medical research team to reach out other patients, but maybe if they could have provided us with contacts of other patients. I know there's a privacy issue, but I think that if we think of it like a consent process, then more than leaving it up to patients, that in our case came from another country, so we didn't know anybody to whom to ask, to provide also the platforms where we could actually have these conversations with the medical team, but also with other people. And there's also another important thing regarding the consent is that the process is an ongoing process. I was just asked last week to sign a reconsent form, and it changes in the protocol these 30 days of high-dose steroids for 60 days of high-dose steroids. When my son Franco was during this phase of 30 days with high steroids, it was absolutely not only a terror, but he became kind of a monster. He was climbing the walls. He couldn't even speak because he was so maniac that he couldn't articulate words. I really cannot imagine him to be at school, because the first 30 days we have to be at the trial site, but then he will have to spend other 30 days in high-dose steroids at school. I don't think he will be able to be sitting in a chair for more than 2 minutes and not kicking or hitting his peers or the teachers. So there's also this fact about the reconsent and how it is done and how it is explained. I would have much appreciated if I would have received more information from the sponsors and the medical team of why these changes are made and what's the rationale of making them.
Okay, I know we're over time, but if we could just quickly
So I wonder if on the informed consent there could be a front page that is an abstract and refers to the important pieces that you need to be aware of, and then references back into the document. Because sometimes it's very helpful to return to a particular issue, and if you read a 22-page document, searching for that sometimes is really difficult. But if it had number 3 as this concern, and you will find that on page 17, I think it might make it easier for a patient or family to be able to identify those issues that they are concerned about over time. And especially in a reconsent, if you say point number 3 on page 17 changed, not that you aren't going to read the whole document, but at least it highlights what to look for, because in reading 22 pages, sometimes you read it and read it and read it, and then you forget what those words mean completely.
just let's go really quickly with the last
Al
two.
McGuire, University of Pennsylvania. I'm just kind of curious the perspective of investigators and parents on the concept of an assent. In children in our study, I was tasked to assent children who were felt to be old enough to have cognitive ability to understand what the trial was about. And I wonder if at times that's at odds with what a parent wants and how people handled that, and both from the investigator and the parent perspective.
I can just comment in our experience. These little boys that are 4 to 7 years old do what their parents say. And as long as their parents are positive and hopeful, I don't think they actually have an opportunity to, I think they really take care of their parents, and they don't have an opportunity or a willingness to say, listen, this isn't for me.
Well, actually, so assent is not generally applicable until a child is around the age of 7 and is able to understand at least the overall concept. And so a 4-year-old would not be asked for assent. But in my experience with the older children in SCID gene therapy trials, if they don't want to do it, it's not going to happen. And on the other hand, by explaining the availability of this experimental treatment, I think that the children themselves have been extremely enthusiastic about it. I think, they probably, like many parents, assume the best, that they are going to get a good outcome. But in any case, we do try to explain it might work, it might not work, we're not sure, and then they decide, and we take that very seriously.
If I had to say, we had our son in a trial, and that was one of the hardest things for us to do because he was about 13, so certainly old enough, to understand. What made it a little easier that it wasn't as critical a trial as, say, gene therapy or even bone marrow transplant. This was a drug therapy, and we knew it was added blood. But that's something that I don't think is really discussed, the effect on the parents, because we didn't know, but we ultimately wanted what was best. And he was going through a lot of pain. And so he basically said if this could possibly help, he wanted to do it. And so that was, but.
Rosemary Hunziker, and I'm an independent consultant now. The stories of the patient and family experiences are extremely compelling, and you guys are very thoughtful and have thought a lot about the things that you think might be amenable to adjustment in some of the clinical trials. So what I'm wondering is, but that bumps up against the fact that the trial design has to be propelled by the ability to achieve some kind of statistical significance so that the results are communicatable and believable to all of the community. So what I'm wondering is, I know you have good access to the people who are sponsoring and developing the trials, but have you had a chance to chat with the statisticians who are also involved in this decision-making process so that you can bring some of your concerns to them, and maybe together there can be some, mathematics, we always think of it as a sterile set thing, but it really isn't. There are some places where you can look at things freshly, and I'm just wondering if you have had conversations with that part of the scientific community to see about any wiggle room or adjustments.
No, I have not, but that is a great option to look at. I have spoken on another panel with patient enrollment at NIH just to let them know how much research I had to do on my own just to find out about some of these trials, and just looking at ways of how to get that communicated to populations who don't have access.
And I'd say we had tremendous access to that statistical community in various ways. One is in which we really have used statisticians to fortify and analyze our natural history study, our natural history database. And we've recently cooperated and collaborated with the FDA and the National Organization for Rare Disorders to populate our natural history study even further by adding placebo-arm data from clinical trials. And there are very fine statisticians in that program that are helping us analyze that natural history and helping us analyze each of the outcome measures that we've selected, showing which ones of those are most potent in terms of statistically demonstrating efficacy based on those endpoints. So that statistician community is very important to us.
And we've been working with the Critical Path Institute to develop a disease progression model. I think our difficulty and our challenges is in these 4 to 7-year-olds in Duchenne, once you start a steroid regimen on them, they improve. So it's often called the golden years of Duchenne, in that 4 to 7 range, where they naturally improve based on refinement of small and large motor function. So we do have some outcome measures that have been validated and tested. And again, we have the complexity of little boys who are distractible. I think this is the difficulty of our population right now, is to really understand what the microdystrophin does in terms of protecting muscle function and slowing down all of the other processes that occur, and trying to measure that quantity, the quantification of dystrophin, and then trying to understand what that means in terms of disease progression. So I think, again, we have to say early days, and also in a disease like Duchenne, in 12 months, you really, in 4 to 7, they're pretty stable. So, they may be doing things better, but they might anyway be doing things better. So it's hard to tease out, is this their normal trajectory? Is this a microdystrophin that is benefiting them? We are also looking for a protein signature and trying to see if we can really identify what the proteins are doing, and if they're changing and normalizing, then we might have a better handle on this.
Following on Pat's idea, I think that for the Duchenne community, it's very difficult to comprehend not only the science, but also the lack of standardized outcomes. One company is using one method to measure dystrophin, microdystrophin expression. Another one is using another method. What are they comparable? What does that mean? Other than, of course, speaking to statistics people from that field, I think that for the patient population, it would be really of help that at least equivalent gene therapy clinical trials speak the same language, so that we can really make an informed decision.
Okay, so thank you for being patient with getting to lunch, but please help me thank the panelists for this wonderful session.